Hi, my name is Pankaj Sharma and I am the Managing Director and co-founder LeadInvent Pharma, a company based out of the US with India R&D Center. At LeadInvent Pharma, we are focused at developing methodologies and treatment protocols for tough to treat cancers. And one of the cancers that we are focused on in terms of product development has been brain cancer, or glioblastoma multiforme, which is a grade 4 cancer in brain cancer. And we have used nanotechnology and photodynamic therapy to come up with a unique product that would be extremely useful for the neuro-oncology surgeons to remove tumor that they are not able to remove using the conventional surgical methods that are being deployed right now. Brain cancer drug development is a pretty tricky thing to wrap your heads around, because brain cancer, it's just not one disease. There are many. It's a multidimensional tumor, but also just the way the tissue is. Brain is a very sophisticated organ in the body, and evolutionary it has protected itself pretty well inside the body. If you look at it, a brain has a blood-brain barrier, which makes it extremely difficult for drugs and small molecules to pass through from systemic circulation into the brain tissue. And that has been a classical challenge in terms of developing a new drug that could be efficacious, which could be systemically injected, and it could reach to the brain tissue or the tumor site. The other big challenge is that most of the brain cancers, they advance very, very rapidly from benign stage, which is a non-cancerous or an early cancerous stage, into a highly recurrent grade 3 and grade 4 tumors. And it's extremely tough at that point of time, because the cancer cells in grade 3 and grade 4 are highly recurrent, highly resistant to chemotherapy and radiation. And the big challenge that we all face at the clinic is that although surgery is the best method to kind of take care of the brain cancer tissue, but it's extremely difficult and sometimes extremely challenging to remove the 100% tumor. In fact, in almost all the surgeries, the surgeons are never able to remove 100% cancer tissue out. And what gets left behind is the seed of recurrence. And in most of the patients, within 5 to 6 months post their first surgery, the cancer comes back and becomes challenging from that point onwards. So at LeadInvent Pharma, when we looked at the problem, we looked at it slightly differently. We wanted to see if we can develop a product that would allow the surgeons to remove the tumor that they are not able to remove using conventional surgical methods. And I'm happy to share that we have been able to achieve a new product, which is a gel, which we call the GB gel. And GB gel is designed to be used by neuro-oncology surgeons during the surgery. So the way that the product has been designed is, once the surgeon removes the tumor out and the resected cavity is available in front of them, they can apply the GB gel much like a spray. And GB gel is designed such that cancer cells will quickly absorb the gel, and the normal cells really don't absorb it that much. And once the cancer cells are loaded with the GB gel, the surgeon will take a low power laser, which is very safe laser, clinical grade laser, and illuminate it into the cavity. The gel does not kill cancer cell. It waits to be excited by a low power laser, and only when a low power laser excites the gel, which is sitting inside those cancer cells, the gel is able to kill those cancer cells very efficiently. So this is like a very precision way of removing cellular level tissues, which the surgeons are not able to remove using standard scalpels and other things. The big thing about this is that we've done cellular data and animal data to understand how good this product is, and I'm happy to share that GB gel is able to kill not only the primary glioma cells, but GB gel also kills the highly resistant and chemo-resistant and radiation-resistant cells. We've also gone ahead and done proof of concept studies in animals, and our data clearly shows that animals treated with a single application of GB gel, they showed complete elimination of their brain cancer with zero recurrence. And because the gel is applied directly onto the site of action, we bypass the chances of systemic toxicity, and also we don't have to worry about the blood-brain barrier. And the amount of drug that we use to excite, or the amount of drug that is loaded inside the gel is substantially lower than it would have been given systemically. So it's a fantastic way of applying the product directly onto the tissue, and it's been designed to work as part of a surgical consumable and surgical drug. We are very excited about the product. We are very excited about the data. We are in advanced conversation with FDA at this point of time. Our pre-IND documents are in conversations with FDA, and we are figuring out the details with FDA in terms of what kind of regulatory studies we have to perform before we can start a human clinical trial, which we estimate somewhere around 12 to 18 months from today. In terms of market, GBM or brain cancer market is a big market. It's about $5.2 billion market, and it's been growing at close to about 10% CAGR year on year. And in the US alone, about 35,000 patients seek grade 4 related surgeries. Kind of give you an estimate as to how prevalent this particular indication is, which means that the FDA is extremely open to enter into conversations for fast approval and quick review of programs which are addressing high unmet medical needs such as brain cancer. And also because it's under orphan indication, companies like ours get an opportunity to move the program much more efficiently through the clinical trials, because we, under an orphan indication, you can move the clinical trials with lesser number of patients, and you can speed up the process so that the innovation moves to the patient in the quickest and the fastest possible way. Also economically quite viable to move it through the clinical phases. In terms of our business model, our business model is very clean. We want to develop this product all the way from an idea stage up to phase 1B, and then we will partner with the right partnering company to further develop and take it to the market. In terms of competition, all oncology indications are hotly contested. It's fantastic to have a program in any of the indications which is moving forward with clear indication in preclinical and clinical stages. What we are excited about is that most of the products that are being developed at this point of time in the market or in the clinical development phases are in post-operative care. So they are drug molecules or radiation protocols which are downstream to surgery. And since our product is part of the surgery, we are upstream to the competition at this point in time. In fact, the way I look at it is that GB gel is going to be synergistic with most of the products that are in development, because if you improve the surgery, the downstream product look definitely better. All of this has come out of an angel round that we did at the start of the company, and we have entered into a fundraise, which is our first Series A round. And I'm happy to talk to individuals and funds which are in the right space, and we have the right map with them. I'm also very excited to share with you that the team that's shaping this entire company up is a stellar team. I'm fortunate to have my management team with us. The management team consists of industry leaders and entrepreneurs with 20-30 years of work experience in drug discovery and development. LeadInvent Pharma is backed up by a strong advisory board, both at the scientific and also at the business side, which brings us a very broad spectrum view and experience to the developmental program, all the way from Harvard Medical School to Novartis to GSK, to some of the other leading companies, and people who have been in those companies are fortunate to be part of the team at this point of time. I am very happy to be talking about this program to all of you, and we have prepared a small product video which would give you a very quick view about GB gel, technology behind GB gel, what makes it really special, and this will give you a very quick deep dive about the product and its potential.
Brain cancer is a tough disease, and one of the most aggressive types of brain cancer is called glioblastoma multiforme, or GBM for short. The tumor is generally radiation and chemo resistant. It's also the #1 cancer killer in children. And only 3% of patients survive beyond 5 years. This disease puts a very high emotional and financial burden on the affected family. LeadInvent Pharma is developing a breakthrough treatment modality that will improve GBM patient survival via a uniquely designed nanotechnology-based gel called GB gel and photodynamic therapy, or PDT. When a photosensitizer is exposed to the laser, it produces hyperreactive oxygen called reactive oxygen species, or ROS. ROS kills rapidly cycling cells like cancer cells by multiple pathways. ROS is powerful enough to kill a 3D cancer cell colony also. Although PDT has received FDA approval for some oncology indications, its use has been limited because of inefficiency in treating large tumors, limited light penetration to treat deep-seated cancers, and skin photosensitivity, requiring patients to stay in dark or semi-dark surroundings, sometimes up to 4 weeks. LeadInvent Pharma's GB Gel overcomes these challenges while retaining the benefits of PDT. GB Gel carries smartly designed nanoparticles, which are approximately 75 nanometer in size. While small in size, each nanoparticle is loaded with multiple photosensitizer molecules. When the laser light hits these nanoparticles amplify the effect of encapsulated photosensitizer and results in enhanced ROS generation. The major problem in GBM is that the tumor is embedded in the normal tissue, which makes it extremely difficult for the surgeons to completely remove the tumor without causing substantial damage to healthy brain tissue. After GBM surgery, generally, a tumor margin gets left behind. Some of these leftover cancer cells are resistant to follow-on chemo and radiation therapy, causing rapid regrowth in 90% of patients within 6 to 7 months post-surgery. And hence, survival time has remained at 15 months even after the best of treatments. GB Gel can help surgeons remove these leftover cancer cells uniquely and safely. After removing the maximum tumor, the surgeon would spray GB Gel into the resected cavity, depositing a fine coating of nanoparticles. Nanoparticles then permeate further, covering up to 3 centimeters area around the site of application, and are preferentially absorbed by leftover cancer cells. Next, the surgeon would place a fiber optic in the resected cavity and would deliver a lower power laser which will activate the GB Gel inside the leftover cancer cells, killing them through ROS generation. GB Gel has been extensively tested on different GBM cancer cells, such as primary GBM cancer cells, chemo-resistant GBM cancer cells, and radiation-resistant GBM cancer cells. GB Gel not only kills primary GBM cancer cells, but also chemo and radiation-resistant GBM cancer cells. GB Gel eradicates brain tumors in animals without recurrence. GB Gel kills chemo and radiation-resistant human glioblastoma cells. GB Gel is made from FDA-approved components. Localized application of GB Gel minimizes chances of systemic toxicity. GB Gel is patent protected. LeadInvent Pharma is committed to harnessing the power of nanotech and laser for eradicating brain cancer.